Biological aging is often perceived as a mandatory thermodynamic decay, an inevitable breakdown that eventually claims every complex organism. For humans, mortality follows a rigid mathematical pattern. An adult's risk of death doubles every 8 years. Compared to the Aldabra Giant tortoise, we are a biological outlier. Tortoises maintain a near-constant risk of death at just over 2% per year, regardless of age. This lack of exponential decay means that biologically, these animals show almost no signs of senescence as they age. The existence of organisms that defy the standard human mortality curve suggests the pace of biological breakdown is a setting dictated by an organism's own internal rules. If these rules are governed by biology, they are susceptible to biological intervention. Mapping the mechanisms that allow a tortoise to maintain its health over centuries provides the blueprint for manipulating our own timeline. Evolution prioritizes reproduction over longevity. In the wild, where external threats like predators and disease are constant, an organism must pass on its genes as quickly as possible before it is killed by the environment. This creates the disposable Soma theory. Once the reproductive window closes, the evolutionary pressure to maintain cellular repair systems drops, allowing molecular damage to accumulate unchecked. However, a discovery in the 1930s revealed that this rate of decay can be slowed. Researchers found that restricting a subject's diet to near-starvation levels can increase its lifespan by a third. Starvation signals a hostile environment for reproduction. In response, the organism activates a survival mechanism that diverts energy away from growth and into an intensive suite of cellular preservation and repair. This demonstrates that the capacity from near-perfect cellular maintenance is already present in our biology. It is simply deprioritized by the evolutionary need to reproduce. Accessing these dormant repair pathways requires identifying the specific genetic instructions that regulate them. In the 1990s, Cynthia Kenyon used the nematode C. elegans to prove that the rate of aging is under direct genetic control. These roundworms are the standard model for longevity research because their simplified genetic structure allows scientists to isolate individual drivers of decay. Kenyon discovered that modifying a single sequence, the DAF2 gene, caused the worms to age at half their normal speed, effectively doubling their lifespan. The DAF2 mutation forces the organism into a state of high resilience, where the cell's internal machinery focuses on repairing DNA and resisting pathogens rather than rapid growth. While humans possess these same conserved pathways, altering the genome of a living adult is a permanent and highly complex intervention that remains out of reach for broad clinical use. A more accessible target is the epigenome, a system of molecular tags that sit on the DNA and dictate which genes are active. As we age, these tags experience epigenetic drift. The molecular markers become disorganized or lost, causing the cell to lose its original functional identity and succumb to the errors we associate with aging. Because enzymes lay these tags down through reversible chemical reactions, this drift can be corrected without changing the underlying DNA sequence. This correction is achieved through epigenetic reprogramming, which utilizes 4 specific proteins to restore the cell's youthful instructions. Dr. Shinya Yamanaka identified that introducing these 4 factors can prune away the chemical errors of the epigenome, restoring the cell's regulatory state. Exposing an old cell to Yamanaka factors physically reorganizes its molecular tags, pushing the cell back to its youthful functional pattern. This suggests that reversing the effects of age is possible by rebooting the software that controls our genes, rather than attempting to rewrite the genetic hardware itself. While genetic therapies mature, we are developing drugs that chemically trigger the body's repair mechanisms without the need for actual calorie restriction. One primary target is the senescent cell. These are cells that have reached their division limit but remain in the body, secreting inflammatory signals that damage surrounding tissue. A class of drugs called Senolytics, such as dasatinib, are designed to selectively clear these inflammatory centers, effectively cleaning the tissue of age-related debris. Metabolic modulators represent a second front in this toolkit, with the diabetes drug Metformin showing significant promise for longevity. Metformin reduces the body's sugar production, which dampens systemic inflammation, and mimics the molecular signals that trigger starvation mode cellular repair. Similarly, rapamycin alters how cells sense nutrients. directing them to break down and recycle the accumulated junk proteins that clog aging cells. Supporting these processes often requires NMN, a precursor to the vital coenzyme NAD+. NAD+ is the essential fuel for the metabolic pathways and DNA repair enzymes that these drugs are attempting to reactivate. Without it, the cell's defensive response is stalled by an energy deficit. This emerging toolkit treats aging as a specific cascade of metabolic errors that can be pharmacologically intercepted and corrected. Applying these molecular interventions on a global scale would result in a massive demographic transformation. The goal is to extend the human health span, the period of life spent in optimal physical condition. This squares off the mortality curve, where the risk of death stays low for much longer before a brief final decline. This health span dividend results in a population that remains biologically active and independent well into their 10th decade, drastically reducing age-related disability. The economic shift accompanying this change is estimated in the trillions of dollars. Financial data shows that the medical expenses for a centenarian in their final 2 years of life are only 1/3 of the costs incurred by those who die at 70 from age-related chronic diseases. The shift from investigating the molecular causes of aging to actively manipulating them has transformed longevity research into a problem of applied engineering.